oh, they did a motor neuron last year. Probably not going to do it. That said, here's what a motor neuron is.
Hi, Monia.
Hi, Maria.
See you later, Amir.
Bye. Bye.
Oh, these
You got a nucleus. These are dendrites. This is the axon. On the axon, there are these things called myelin sheaths. They basically act like they're the same thing as why you have insulating wire on an electric wire. These basically help you to send a signal faster down your axon, which could be as long as like 3 feet.
Oh
Wow.
my god.
That's long.
That's the nucleus. Here or the synapses.
So is that why it will take a while to react to jokes?
That's the motor end plate, and then the places in between.
And
Oh, I didn't label a myelin sheath.
That too. Because people don't think it's such fun.
Ranvier. Now, it is worth remembering that there is something that they are probably going to ask you about, and I've tried to speckle this in a few different units going all the way back to our cell membrane unit. There's this thing called the sodium-potassium pump.
Yeah, we did that.
They are everywhere on this cell membrane. And the reason is that they are constantly pushing sodium and potassium onto one side of the membrane in active transport. It uses ATP. It's an energetic process. You push things against the concentration gradient from low to high. So, sodium is usually out. Potassium is usually in. When you have an action potential, what happens is all of the sodium-potassium pumps turn off for a second, and then they turn back on again. And it's a wave of them all, like taking a millisecond-long nap and then turning back on again. And that wave is going to allow, momentarily, the sodium to diffuse in, the potassium to diffuse out, which we abbreviated as synpout.
I remember that.
So synpout is the wave. We practiced this out in the hallway with waves. We had arms out and in, and we did this, and that was the diffusion, and then we had to go back again. So that is called an action potential. And it consists of a wave of what we would call depolarization. Because, and I'm going to actually double check this real quick so I don't give you the wrong information. Which one is positive and negative? I'm pretty sure positive is on the outside, but just let me double check.
Yeah,
Ranvier.
Is that an M over there?
Why a scribble? That's a scribble.
R-A-N-V-I-E-R.
It's a scribble.
It's a scribble.
Person messed up.
Everyone would say it's a scribble at once.
1, 2, 3.
It's a
It
scribble.
makes me really happy.
That was so successful, guys. Good job on that.
That was pretty bad.
Powerful.
You asked this question
Well, maybe if some more people can.
Yeah,
Like,
yeah,
really?
Yep, I was right. Hey, I was right.
What is it
Okay.
referring to? Just
Oh, well, maybe if a group
So,
of friends went to
the
California,
node-
but you know.
The space in between the sheets.
Did you take a shot of vinegar before you came here, Kyle?
No,
Because you
I shook
bitter
a shot of
AF. Oh my god.
Okay.
We got permission.
Last point, folks, real quick. If I was to zoom in on a section of the cell membrane and just look at it as a tube, the sodium on the outside, when the pumps are on, that has a positive charge. The potassium on the inside has a net negative charge. But in the moment, with the actual traveling action potential, this thing is zooming this way, they flip. The potassium diffuses out, the sodium diffuses in, syn, pout, and momentarily it will be like this. And that flip of the positives and negatives is what we call depolarization. So, tying together a few things that we've sort of speckled in across units, just in case it comes up. Who knows? It's such a lottery on what's going to happen here and what you'll actually be asked about.
Yeah, how can you do it in the water? How can you do it in the water? Because somebody else did.
All right. Check the motor neuron. What next?
We do cell membrane.
Condensation.
I'd say phospholipid bilayer.
All right, let's do the cell membrane quick. That'll be a good way to review a few of those ideas.
Most of it, but I just don't remember the names.
Nitrogen.
Oh, that's
All right.
what
Here we go.
I
We're doing the
pushed.
fluid mosaic model.
This takes really forever.
We've got a double layer of phospholipids, which we call the phospholipid bilayer. You don't want space between them. You also don't want too much space between the tails. We'll label a bunch of this stuff here in a moment. You should have a protein that represents a channel. And a channel would basically just be a tube. An opening that is large enough for things that are too big to squeeze between phospholipids to just travel through the membrane anyway. A couple more phospholipids. And there's a few different ways you could show this. I'm just going to do it this way. It's not really super important what shape you give like a pump, but that'll be my pump. There are these proteins that exist only on one side of the membrane. They are called peripheral proteins. Off of the peripheral protein, you might have a receptor. Could look like a Y. And that receptor might be what a virus binds to be welcomed into your cell via endocytosis. In addition, inside, I would put a couple of hexagons that will represent cholesterol. Cholesterol is a saccharide, it's a sugar, so it's appropriate to show it as a hexagon. So that's the drawing. Those are the pieces, but what the real trick is knowing why and how everything moves through these things, which I'll talk about here in just a moment. Yes, Amelia?
I remember when we first did this that you said we could also have the lines instead of having this little not triangle, but you know what I mean.
These little lines?
That we could just have them straight instead of an angle.
Yep. And if you were doing it really, really perfectly, one of them would be bent. But it's not a big deal.
Is that one supposed to be touching? The end, on the very end?
No, they
Okay.
could be touching.
Okay,
It's
then
not. That's
it's
not
not.
an intentional thing.
Oh, okay.
It's supposed to be like that. Yeah, Sarah.
So if you're drawing a diagram, like say we were drawing the motor neuron, and I'm not going to remember all the names for everything, would you get any marks if you draw a structure but don't label it, or do you have to label it to get like a point on it?
Usually, it's points are given for the labels that are correctly given.
Okay.
So drawing it without labeling it probably wouldn't yield any points. All right, let's label the crap out of this thing. These guys, I'm going to do this in a different color. These guys right here are called the phosphate heads. And they are hydrophilic. These are the fatty acid tails. And they are hydrophobic. Which explains why the layer forms. If you remember, the fact that one side is, actually I'm going to undo that. Over here, the head is polar, the tails are non-polar. Polar things like water. So if you're polar, you're hydrophilic. Non-polar things don't like water, they are hydrophobic. When you have both of these properties in the same molecule, that is called being amphipathic. So the amphipathic properties of phospholipids mean that in water, they will form a bilayer naturally. The tails will be repelled by water, the heads will be attracted to water, but the tails will be moderately attracted to each other. So they form this bilayer. This is cholesterol. It is worth noting that that is only present in animal cells. It is a stabilizing molecule. It helps provide a little bit of sturdiness or stability to the membrane. This is a protein channel, or the Chanel protein. This is a protein pump. This is a peripheral protein. And this is a glycoprotein receptor. Glyco, because each of these little circles is actually a hexagon, and they're actually all sugars. So, there you go. Now, a quick review of how all of these things work. If you are a really, really small molecule, you can move in between the phospholipids and get from the other side, from high to low, in the process of diffusion.
I lied.
Only if you are small, and only if you are non-polar. If you are small and polar, like water is, I'll draw a little Mickey Mouse here, you might be able to get through the membrane, but the fact you are polar means you don't do very well here. There's actually a repulsion from you getting through. So water more likely moves through these channels, but it can squeeze through the membrane, just not quite as easily. But water moving through the membrane is not called diffusion. It's called osmosis. And it moves from where to where?
High concentration.
High to low concentration of water, but that's not what you want to talk about in osmosis. You want to talk about the solute concentration. And if there was a lot of salt down here and not very much salt up there, this is called hypotonic. That is hypertonic. Sort of like hypothermia is a low temperature. Hypotonic is a low solute concentration. Hypertonic is a high concentration. You can think of hyper like if there was a lot of sugar, you would feel really hyper. When the two are equal, if they ever equalize, you have two solutions that are equilibrium is for diffusion. Isotonic. If they are the same, they are isotonic. And so water moving from hypo towards hyper until they are iso through a membrane. There must be a membrane. Now you're talking about osmosis. Yes?
What about integral proteins?
Both of these, thank you, actually, we can add that. They are both integral proteins because an integral protein is simply any protein that is in the membrane, basically top to bottom. If you are doing a protein channel process, you are probably talking about facilitated diffusion, which is the exact same thing as diffusion, except it is helped or facilitated by this big hole that allows something that is a little bigger than normal, something too large to fit through, not going to be able to squeeze between the phospholipids, so it goes through the channel, again from high to low. And then you've got the protein pump, which does active transport. And this is the process that breaks all those rules we made before. Every one of these processes was things spreading out. Active transport crams things together from low to high concentration using the power of ATP. Yeah?
Realistically, if we get a question asking us to diagram this, how many marks is it gonna be worth? Because I know like polar and non-polar would be considered like 1 mark, but like if you were the one who wrote this question, how many marks would you probably?
6, 8, I don't know. Honestly, the number of marks really only tells you how much time you should spend. Because you should write everything you know.
Right.
And the only limit to how much you write, hopefully, is the amount of time you have to write it. So, my guess is that on paper 2, most people are going to finish early. Most people are going to take the whole time with paper 1, maybe a few finish early. Folks usually finish a little early on paper 3 as well. So, if you have extra time, go back and add all the details and all the labels and all the annotations you can. With the exception of being self-contradictory. That's the only time that hurts. If you really don't know how to answer a question, do not say, enzymes bind with active sites. Also, enzymes bind with fruit cups. It's like, well, you've made 2 statements that cannot be accepted together. They're contradictory, and even if one was right, the other one's wrong, and they're not going to take either of them. Or perhaps a more realistic example of what I just said would be like, phytochrome far red versus phytochrome red. Which one is it? Don't say it's one in one spot and it's the other one in another spot. They won't take either mark at that point. Okay. That was a laugh. Done. Done with the fluid mosaic model.